Hey, hi. So in this video we will see about glycolysis and its alterations in RBCs. So glycolysis is this. So glucose enters from the blood into a cell through a transporter called GLUT2 or glucose transporter. Once it enters a cell, glucose undergoes a first primary metabolic pathway is glycolysis to form pyruvate. And this pyruvate subsequently enters into mitochondria and which will be converted to acetyl-CoA, which enters into TCA cycle to produce ATP. However, this is not what that happens in all the cells. That is, oxidation of glucose beyond pyruvate depends upon two factors. One, availability of oxygen. Second, availability of mitochondria. How? For example, in the presence of oxygen, the end product of glycolysis is always pyruvate, which is further oxidized in TCA cycle to form ATP. This is called aerobic glycolysis. However, in the absence of oxygen or in the cells which lacks mitochondria, glycolysis leads to formation of a compound called lactate. That is, there is no oxygen, there is no mitochondria. So what happens to this pyruvate is it gets converted to lactate by an enzyme called lactate dehydrogenase, and this is called anaerobic glycolysis. So those cells which lacks mitochondria, the product of glycolysis is not pyruvate, it is lactate. And this lactate will be converted to glucose by gluconeogenesis for their energy demands. There is one more important difference in glycolysis in RBC, red blood cells. That is, there is an alternative pathway for glycolysis in RBC which is called as Rapoport Leubering shunt. And there's one important product that is derived from this shunt, which is called 2,3-bisphosphoglycerate, which is also known as 2,3-BPG. So what is this pathway is about? See, in a normal conventional glycolytic pathway, that is the glycolysis happens in the normal cells, glyceraldehyde is converted to 1,3-bisphosphoglycerate by an enzyme called glyceraldehyde 3-phosphate dehydrogenase, and this 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate. This is what happens in cells other than RBCs. What happens in RBC is this 1,3-bisphosphoglycerate is converted to a compound called 2,3-bisphosphoglycerate or 2,3-BPG by an enzyme called bisphosphoglycerate mutase, and this 2,3-BPG is converted back to 3-phosphoglycerate, which enters into normal subsequent glycolytic pathway. So what is the importance of this compound called 2,3-bisphosphoglycerate? This 2,3-bisphosphoglycerate is actually a compound which can reduce the affinity of hemoglobin towards oxygen. That is, this 2,3-BPG goes and binds to hemoglobin. Upon binding to hemoglobin, hemoglobin will lose its affinity towards oxygen. Thereby, oxygen unloads itself from the hemoglobin. Okay, so that's why you see 2,3-BPG levels are high in those people who are living at high altitudes. That is, they cannot afford to bind oxygen tightly. Right, because in high altitudes the partial pressure of oxygen is reduced, so they have to let go of the oxygen whenever it is in demand. So this 2,3-BPG is one such molecule which can reduce the affinity of hemoglobin towards oxygen. To summarize, red blood cells lacks mitochondria and they are completely dependent on glucose as metabolic fuel, and glycolysis in the RBC is mostly anaerobic and the product is usually lactate. Unlike other cells, glycolysis in the RBC produces an intermediate called 2,3-BPG, which upon binding to hemoglobin favors unloading of oxygen in the tissues. So that's it. I hope it was useful. Thank you.